Thank you very much, Alfredo. I think that it's clear that if we're going to understand blepharospasm, we have to understand all of the different factors going from the environmental influences, genetic influences, and how they cause blepharospasm. And the physiological studies that Professor Berardelli showed us are the way that you can analyze what blepharospasm looks like in terms of what the patients show, that these are the different findings that lead to these spasms. And somehow we have to link them together. How do we get from the environment and the genetics to the physiology to the actual manifestations? Now, and so that's the sort of logic of how we're making these presentations. And one of the other ways that one's able to link this information together that has always been helpful in various illnesses is to look at animal models. And animal models have not been very common or very good in the focal dystonias. But we have, fortunately, one of the leaders here in that field, Craig Evinger, who has been interested in blepharospasm for many years. He's professor of neurobiology, behavior, and ophthalmology at the State University of New York. Has been really producing some very interesting work, which you've heard about already from Professor Berardelli, and I'm sure he has some more interesting things to tell us about. Craig.
All right. So thank you very much for the Blepharospasm Foundation for hosting me and for Dr. Hallett for inviting me. It really is a pleasure to be back speaking to you again. And what I want to, again, talk about are animal models and what they can and can't do for us. And you think about, what do you want in an animal model? And it seems like a simple question, but it's really not. We've had some success at creating animal models of blepharospasm, and I'll talk a little bit about why the reason for that is. But there are lots of animal models that have been more difficult. So, for example, Dr. Ozelius told you about the DYT1 or the TOR1A mutation. And you can create that mutation in mice without any great difficulty, but those animals don't show generalized dystonia. Now, that doesn't mean that animal models aren't useful, right? Animal models are part of the process we really need to understand blepharospasm. We need the studies like Dr. Ozelius has been talking about of understanding what are the genetics, what are the predisposing factors for blepharospasm. We need the epidemiological studies that Dr. DeFazio has been doing. What are the risk factors? What's going to lead to the creation of people developing blepharospasm? We need the really insightful observations that you've heard from Dr. Berardelli, Dr. Jankovic, about what are the conditions in blepharospasm and how do they relate to other forms of dystonia, the pathophysiology. And from all this kind of information, we create all sorts of hypotheses about what causes blepharospasm. And this is where the animal models come in, because we can use animal models to test some of these hypotheses. And so today I'm going to very briefly describe some of the information we've got from animal models in the past. Then I want to talk why animal models work well in blepharospasm. And finally, I want to finish by discussing how we're using animal models to investigate what Dr. Berardelli described very nicely as metaplasticity. I'm beginning. After this here, you hear this talk, you're going to be thinking I'm tracking Dr. Berardelli in the snow here, but you'll see it's very similar. So first, some of the animal models. So we've had at least two things that have come out, interesting things that have come out of the animal models. One is we used this predisposing factor that you've talked about, and in the case of creating this animal model, we used a change in dopamine, one of the things that Dr. Berardelli and Dr. DeFazio talked about, and we combined that with a dry eye sort of factor. And when you combine those two, you can create an animal model that has blepharospasm-like spasms of lid closure. Okay? So we know that it is possible to create blepharospasm with these two kinds of factors. Another one we've been looking at, we and other people, are photophobia. So photophobia is the pain that you experience with light, and it's very common in blepharospasm patients. Now, think about what happens when light enters your eye. Right? So light comes into your eye, it activates neurons in your retina, and the other thing it does is dilate blood vessels in your retina. Now, the reason for that is your neurons are real energy hogs. Right? Everybody's neurons they're the Humvees of the body. Right? When they do anything, you have to increase the blood flow to make them keep up enough energy for them. It turns out that the trigeminal system, which Dr. Baradelli and other people have talked about, which provides all the sensation for your face and inside your head, has pain receptors or nociceptors that are attached to these blood vessels in your retina and blood vessels in your brain and your dura and the covering of your brain and all sorts of places like that. Now, in people who have blepharospasm where the trigeminal sensitivity is really ratcheted up, when light comes in your eye, you get dilation of the blood vessels, and those nociceptors or those pain receptors become activated, and you sense that light is painful. So, in fact, in photophobia, as you're going to hear much more about from Dr. Katz today, at least part of it is you're not responding directly to light. What you're really responding to directly is dilation of your blood vessels. So in that way, you can think about photophobia as being like having a migraine in your eye. Okay? So those are some of the things that have come out of this. And what I want to focus on today, let me go on and tell you why animal models work in blepharospasm. And that is because all God's mammals blink the same way. Right? There are a few little differences, but basically they're all the same. And you've already seen some examples of the orbicularis. Right? Here's the orbicularis in a person. It's this sort of circular muscle here. When it contracts, it's like pulling purse strings. Closes your eye like that. Here's an example of the same thing in a guinea pig. Okay? It's exactly the same. When you blink, I'll get this right yet. Okay. When you blink, it's the same sort of way. So now Dr. Baradelli showed you some of these things. So here we electrically stimulate the supraorbital nerve. It feels sort of like someone taps you like that. Okay? Then here's the electrical activity of the orbicularis muscle. You're going to see a lot of these. My wife, who's a molecular biologist, always says, you aren't going to show more squiggly lines, are you? And yes, we are going to see squiggly lines today. But this is the electrical activity of the orbicularis muscle closing. And when it contracts, it pulls the eyelid down, and when it turns off, the eyelid comes back up, a pretty standard blink. And you can see exactly the same thing in a guinea pig. Here we stimulate the supraorbital nerve, here's the orbicularis, contracting, pulls the lid down. Okay? So the reason it works well for blepharospasm is rodents, all mammals blink in basically the same way. On the other hand, if you start thinking about having focal hand dystonia, writer's cramp and things like that, primates are about the only animals that use their fingers really independently, right? Most animals don't do that. They live in a different world than we do. So blepharospasm and working in the eyelids turns out to be a great system for animal models. So here's what I really want to talk about today, and that is the issue of really metaplasticity, but I'm going to call it homeostasis, and it's really, for the purposes of what we're talking about, it's going to be the same term here. And my hypothesis is that blepharospasm develops when dry eye, the motor responses to dry eye, get out of control. Right? It's as if you're learning, and you do learn to respond to dry eye. When your eye becomes irritated, you change your pattern of blinking, you blink more frequently, you can end up with spasms, as Dr. Savino was pointing out. You can end up with things that look like blepharospasm just from dry eye. And what we want to argue is that in this case, the motor learning becomes dysfunctional. Okay? It is out of control. And so the key, from my mind, to understanding blepharospasm is to determine how and why motor learning becomes dysfunctional. Okay. So what we need to talk about is learning. And so I'm going to step back for a minute. And because I'm a neuroscientist, I'm going to talk to you a little bit about why motor learning, or why learning in general, how we see it happening, and why it's a complicated process. Okay, here's the problem. All right. learning occurs because connections between neurons, which are called synapses, either get stronger or weaker. That's really what learning is. So you can think about learning in sort of two general ways. You have declarative learning, right? You remember when you were married, unless you want to get in big trouble, right? You remember the capital of California, things like that. They're things that you have to think about to remember, okay? Things that you have to think about to learn. Then there's another group called non-declarative memory. These are ones that you don't think about. So like when you ride a bike, you don't think about riding a bike, you just ride the bike. When you don't think about tying your shoes, you just tie your shoes. Your motor system has learned some things, and you don't have to consciously think about them, and that's what we're going to be talking about. Now, all these forms of learning involve changes in the strength of synapses. So why is this a complicated problem? Well, look at it this way. So here's your brain, and despite what your spouse tells you, all brains are basically about the same size. Okay? Now, in each brain is composed of individual neurons, and there are 100 billion neurons in your brain. Now, we've been talking about big numbers, right? And this is very important now that we're thinking about the economy and $1 billion here, $1 billion there. So what does a billion really mean? Okay, so here's a simple way to think about it. Everybody knows how long a second lasts, right? So to live 1 million seconds takes about 11 and a half days. That's how long it takes you to live 1 million seconds. To live 1 billion seconds takes you 31.7 years. Right? So there are a few of us in the room here who've lived 2 billion seconds. There's almost no one here who's lived 3 billion seconds. So what are we talking about for 100 billion? We're talking about living for over 3,000 years. Right? So we're almost 1,000 years past the age of Christ, right? For the time of Christ. Now, each neuron has somewhere between 1,000 to 10,000 synapses. So that means all these neurons are connected to at least between 1,000 and 10,000 other neurons. What that means is that you have somewhere on the order of 100 to 500 trillion synapses. Okay? Any guesses how long it takes to live 500 trillion seconds? Almost 16 million years. Right? So that's longer than people have been on the earth. All right, that's why the problem is complicated. So for neuroscientists, who are basically a very simple-minded group of people, we start thinking about it in a simpler way. We start thinking about it for single synapses. And this is going to get back to the LTP that Dr. Baradelli talked about. So here's going to be a blow-up of a synapse. So what you have is, here's a picture of a neuron. It's sending electrical activity here. And this is called the presynaptic neuron. And it's coming up here, and that's its presynaptic terminal. And it's contacting another neuron, which is we're calling the postsynaptic neuron. And it has the postsynaptic. So you have this connection here, the synapse. These cells aren't touching each other. There's actually a space between them. So what happens is electrical activity comes down here, like this. It causes this presynaptic terminal to release neurotransmitter. And neurotransmitter are just little bitty chemicals. Most of the brain neurotransmitters, at least that excite neurons, release glutamate, right? Very simple stuff. The glutamate diffuses slowly across here, and then it binds to a group of neurotransmitter receptors. These receptors open. They're sort of like gates. They let different kinds of ions go in. And when that happens, the cell starts responding and sends its signal, and then it will go on to the next one. Okay? So, when we think about learning, we're saying that the synaptic connection gets either weaker or stronger. So let's think about it getting stronger here for a second. One of the rules about how things get stronger in the nervous system is that if the postsynaptic neuron is active, when the neurotransmitter receives it, it gets stronger. This connection gets stronger. So the signal comes in, it diffuses across. This one's active, you get another response. Now what happens here is because this one was active at the same time all this neurotransmitter hit it, there are lots more of these receptors sort of floating around in here. And you just stick more onto the surface here. It's like adding more gates. Okay? So now, when the signal comes in, diffuses across, you get a much bigger response because, went too fast there, because there are more receptors here. Now, there's an inherent problem with this, and this is the one that Dr. Baradelli pointed out. And that is, so I make it a little bit stronger, and the next time it stimulates, gets a little bit stronger, gets a little bit stronger, gets a little bit stronger, and before long, what you have is the system stuck with too many receptors here. It's stuck in the upper end. It can't do anything more. You can't learn anything more from a synapse that can't change. So what your nervous system does, and what synapses do, this is when synaptic learning becomes dysfunctional, they need homeostasis. And you can think about homeostasis, again, as I said, and metaplasticity is the same sort of thing. What you want to do is make this synapse, you want to keep your synapses from getting stuck in this upper range or stuck down in a lower range. And so what you do is you have homeostasis, which has a lot of different mechanisms, but what it does is it keeps these working in the same range. And so you can think about it as that the stronger a synapse might get, the harder it is to add more receptors. Okay? Now, this is sort of your basic neuroscience lecture here. I won't bug you with it anymore. Now I'm going to link it back to blinking. All right. As Dr. Baradelli very kindly showed, so here we have stimulation of the supraorbital nerve. Here's the electrical activity in the orbicularis oculi EMG. To think about how synaptic learning links to learning in a blink circuit, you can think about the SO, the supraorbital nerve stimulation, as being equivalent to the presynaptic neuron here. The orbicularis activity here is equivalent to activity in the postsynaptic neuron. So if you present supraorbital stimulus here, evoke a blink, and then present lots of these high-frequency stimuli here, these supraorbital nerve, it's like releasing neurotransmitter when this one's active. It's going to potentiate the blink reflex. It's going to make it stronger. Right? And this is exactly what you're seeing right here. When you do this in normal humans, you do get an increase usually. This is analogous to that long-term potentiation we talked about occurring in synapses. Here's some work from Dr. Quattaroni, and he's been working with Dr. Baradelli. And in fact, the Research Foundation has in fact supported some of Dr. Quattaroni's research, which has been really elegant, looking at blepharospasm again here. And when you do the same high-frequency stimulation in both control subjects, which you see right here, and subjects who have blepharospasm, you see this. So here are more of these wiggly lines, but here you can see the blink occurring right here, okay? And in a blepharospasm, you can see that's about the same size, right? And this is before high frequency. Then they do this high frequency, and you see you get an increase in the size in a healthy control, but it's much, much larger in the blepharospasm patient. Okay, and this is just graphing showing that sort of thing. So you can think about a patient with blepharospasm as having somewhat dysfunctional learning, right? It's as if they can't control. It's as if the metaplasticity in a blepharospasm patient is fouled up. And so I actually think about it in some ways as if you've gotten too many receptors here when you have blepharospasm. So if you were going to look for homeostasis or metaplasticity, what would you expect? Well, I think of it as very much like the thermostat in your house, right? You know, you've got this temperature. When it gets too cold, the heater kicks on. When the temperature gets too hot, the air conditioner kicks in. You're trying to keep your temperature at a pretty constant level, right? So you can think about the blink reflex in exactly the same way. Your brain has this ideal of what a blink should be. It says, okay, it should be this big, given this sort of stimulus. And so it tries to move it back and forth to keep it in that range. So one way you might think about this is, if you have blinks that are smaller than this ideal size, when you do the high-frequency stimulation we talked about, the learning you should get is an increase, a potentiation of the response. When the blinks are larger than the ideal, then high frequency should make the blinks smaller, right? Because you want to stay in this range. So here's where the animal studies come in. Now, the reason you do this study in animals, and I can tell you, having been in the high-frequency experiments myself, they just aren't interesting. Right? I mean, you experience them, and that's it. And if you told me you're going to have to come do this for the next 40 days, I'd say, no, I'm really not all that interested. Right? But the rats, on the other hand, don't have a choice. Right? So we do the same experiment in the alert rats. They're wandering around their cages, getting the high-frequency stimulation. And first, just to show you that it actually works, more squiggly lines. Here's the stimulation of the supraorbital nerve. And most of the records I've shown you before, we've only been showing you the top half of the response. Here you're just seeing the whole response, but it doesn't change anything. And you can see when you got a small blink like this, after the high frequency, the blink gets much, much larger. Okay, again, no surprise. Now, how can you see homeostasis? Well, what we do is we do this experiment day after day after day. And so we first measure how big the blinks were at the beginning of the day. Then we're going to do the high frequency. Okay? And then we're going to measure, just as you saw with Dr. Baradelli's work, we're going to measure it for immediately after the high frequency, 30 minutes to an hour after the high frequency. And then you can just average those together and see what really happens. And so here's a graph, right? And so you can think about this is the average change relative to what it started at, relative to its first amplitude. Okay? And so when it goes up like this, the blink was potentiated. It's larger than it was before. Down here, the blink is smaller after the high frequency. Blinks are smaller after the high frequency. And here's what it looks like. Each one of these points is a different day. Okay? And what you can see is that when you start out with this amplitude and this 3,000, it's just a number. It doesn't mean anything in general. So about 3,000, you get about a 40% potentiation following the high frequency for an hour. Okay. On the other hand, if you're about 6,000, you get about a 40% depression. You get a reduced amplitude. So what this is doing here is you're seeing this relationship. This is homeostasis. This is metaplasticity. The blink circuit is trying to keep itself within an ideal range, and we define the sort of ideal blink size, and this is arbitrary. You can think about at this point where it crosses the 1. So you would say if the blink was this size, in theory, if a high frequency occurred, you wouldn't get any change because that's the perfect size for the blink. Okay? Now, the other thing that's important here, so we think of this as the set point, like setting the temperature on your thermostat. The other thing is this slope. This slope measures how plastic your trigeminal system is, how plastic this circuit is. How much change can you get? All right. Now, what we looked at before measured this for 1 hour. That's the change. We're looking at it for 1 hour. I mean, if this is really important, it's got to last longer than that. Right? Because obviously you can make all these little changes and they'll change it for an hour or so, but what happens over days? And here again is back to the animal studies where you can look at the same animal over long periods of time. And so what we've done here is measure the amplitude of the blink before the high frequency stimulation. That was the same value you saw right here. Do the high frequency stimulation. The animal spends the day doing whatever the rat does after the high frequency, and the next day you bring him in again, and now you measure what was the amplitude the next day before you gave the high frequency. Okay? And what you find is that when you start off on day 1 with a small blink, and you give the high frequency, day 2, you've got a larger blink now. It's this relationship again. And when you start off with a large blink on day 1 and you do the high frequency, by day 2 you have a smaller one, right? So what it means is this change has lasted more than a day, right? It's lasted at least a day, and some of these, this is all run with undergraduates. I'll explain in a minute, and they don't work on the weekends, right? So there are lots of times when there are 2 and 3 days between these experiments, and so in fact what happens is that these are long-term changes. So this high frequency is producing long-term changes. All right. Now, The issue is dry eye, right? Because dry eye is what the real problem is. So here's another rat, a different rat. And here's the same thing you saw before. Here's the change in blink amplitude after high frequency, and then the blink amplitude that started before the high frequency. And you can see when you have large blinks, you get depression. When you have start off with small blinks, you get potentiation. And you can see the slope of this line, and that would be the set point. And you can see that the next day, that's still there. All right, you still get, these changes are lasting a long time. So now we can take these rats and create a unilateral dry eye. It's not a complete dry eye, but they clearly find it like dry eye. You're taking out part of the lacrimal system, so part of the tears have been taken away. And here's what happens with that. So now you see, first, blinks get larger. And that definitely happens in people. When people get dry eye, their blinks start getting larger, which is no great surprise, because as Dr. Savino was pointing out, that's the kind of thing you're going to do in response to dry eye. You want to improve the tear covering of your eye. You want to blink harder. You want to blink more often, those sorts of things. You still see this relationship between a high frequency, between large amplitude producing depression and high and larger blinks producing potentiation. But the thing that's most striking here is comparing the slopes of these two lines. These neurons, the blink circuit now is less modifiable. It has less plasticity than it did before. Now we're exactly back to what the synapses were at, right? When the synapse starts to get too strong, you make it harder and harder to get it to increase its strength. And that's exactly what's happening here. And you can see that, in fact, it's very hard to change its real strength from over long periods of time. It's really starting to fix itself. Okay? So what we want to argue is that, in fact, blepharospasm is not allowing this sort of reduction in plasticity to come about. In fact, what we would argue is that, given Dr. Baradelli's point, is in fact, if you were to do a person with blepharospasm, they're even going to show a steeper slope here than they did before they had blepharospasm. All right? So for us, the real question now is understanding where this blepharospasm, where the homeostasis or metaplasticity is coming from. And where we think it is a region of the brain called the cerebellum. Okay? And there are a number of reasons to think why the cerebellum might be important. And so here's an example from Dr. Baker, another person that the foundation has supported over time. And these are fMRI images. So they're measuring, what they're really measuring is the activity of neurons, and they're measuring indirectly because what they're measuring is blood flow here. And the more blood flow you see, the more activity you assume is going on. And here's a control subject, and here's a subject with blepharospasm, and you get lots of areas excited. But in particular for us, the emphasis here is on the cerebellum. And so what we're gearing up to do with animals right now is start recording neurons here during this high-frequency learning in this cerebellum and record lots of neurons at the same time so we can start to get some idea of how these changes are and how they're going about. And then eventually what we want to do is take our animal models of blepharospasm and see what happens to the neural activities in these areas. And is this a spot where metaplasticity falls apart in blepharospasm where it doesn't fall apart in control subjects? All right, so let me finish up by showing you a picture of Stony Brook and thanking a lot of the people who've helped. Haripriya, Joanna, Bethany, Michael, and Daniel are actually undergraduates who work in my lab, and Céline Dauvignan and Jianbing Miao were postdoctoral fellows in my lab, and all this work has been supported by the National Eye Institute. Thank you very much.